Okay, so this is the first lecture for chapter 4, where we're going to talk about tissues. So just as a reminder, we talked about this in the first chapter, but tissues are groups of cells that are similar in structure and that perform a common or related function. So individual body cells are specialized. So for example, if we're talking about a skeletal muscle cell, that's very different and specialized for muscle contraction compared to a liver cell. And each type is going to perform specific functions that help maintain homeostasis throughout our entire body. When you talk about studying tissues, that's called histology. So your entire second lab exam is on histology, looking and studying and identifying the tissues. So there's 4 basic types of tissues. We have epithelial, which is typically our covering tissue. We have connective, which is typically our supportive tissue. We have muscle tissue, which is going to help with movement. And then we have nervous tissue, which is going to help control everything within our body. So 4 basic or 4 general tissue types. So it's just a location of where we can find some of these tissue types in the body. And we can talk just briefly about microscopy. Not too much information here. Basically what these terms mean. So if we're talking about fixing a specimen, that means we're talking about preserving it in some sort of solvent. In order to prepare a slide for us to view, it has to be sectioned or cut into thin slices so that we can transmit light through it or electrons through it if we're using an electron microscope. Also, most tissues are translucent, so that we can see right through them. So we have to do some sort of staining, and different types of tissues will stain differently. So that's what leads to the various colors like pink and purples and blues that we can see when we look at it under a microscope. So staining is really just to enhance the contrast. However, whenever we go to stain anything, it's going to cause distortions, and these distortions are known as artifacts. So in A&P2, or I think you looked at it in A&P1, was the artery, the nerve, and the vein. Well, anytime you go to prepare any type of blood vessel to view under a microscope, it causes the veins to collapse. So their lumen is distorted and doesn't look symmetrical or like a circle like it should, because the staining process has caused these distortions. There's a couple different types of microscopes. We use a light microscope, so the staining on your slides are going to be colored dyes, typically from some sort of food product. Electron microscopes require a different type of color in order for them to be viewed, and that's going to be resulting from heavy metals. So this is just a picture showing you the difference. So on the left-hand side over here, this is what cilia look like under a transmission electron microscope. And then to the right, we can see what cilia look like under a scanning electron microscope. Neither of these do we actually use because we use light microscope.